Hi everybody, so welcome to today's Brains, Minds, and Machines seminar. This seminar was organized by the CBMM Student and Postdoc Council, and we're super happy to have the opportunity to have Dan Kersten here for a talk. Dan is a professor of psychology at the University of Minnesota and a member of the graduate programs in neuroscience and in computer science, so a perfect fit for CBMM. His early studies were in mathematics, with his undergraduate here at MIT, and a master's at Minnesota. He then turned to visual perception, completing a PhD in psychology with Gordon Legg at the University of Minnesota and a postdoc with Horace Barlow in the physiology department at Cambridge University. He also spent time as a visiting scientist at the Max Planck Institute for Biological Cybernetics and here at MIT as well. And he has lots of really good old friends here. His work has spanned from Bayesian models of object perception to early neural coding of images. And the students felt very excited about inviting him here today because his conceptual clarity in laying out formal frameworks for vision, all the while with his keen eye towards our perceptual experience, has really inspired many of us studying perception. Today he'll be talking about his investigations into the computational functions of feedback to the early visual cortex. And with that, please join me in welcoming Dan Kersten.
Thanks very much for that kind introduction and for the invitation to come. How's my volume? Doing okay? Good. At lunch today, one of the students asked how I got interested in vision, given that I had a background in mathematics. And I owe it to MIT's definition of what a humanities breadth requirement was back in the 1970s. Because I could major in math or you could major in physics back then, and you could satisfy your humanities requirement by taking courses from Whitman Richards, Dick Held. I took a course from Jerry Letvin. And so neuroscience and brain science somehow qualified as humanities. Without that, I probably wouldn't have been inspired to go into vision later, so I appreciate that. I also owe a big debt to MIT over the years because of the inspiration I've gotten from the many faculty and the students of students over the years who's I probably followed their research more than any other university or college where I have colleagues. So we start off with the basics to orient everybody. I think everybody's familiar with this, at least most people are familiar with the pattern of feed flow of information at a coarse scale throughout the visual system. This is mapped onto the human brain. And later on I'll be mainly talking about what we believe are effects in V2 and V1, early visual cortical areas. And people are probably also familiar with this. This is a version of a almost compulsory visual hierarchical diagram that was used in so many talks over the years from Fellerman and Van Essen. This is a version due to Wallish and Mavshan, which tries to illustrate the relative contributions or the relative proportions of neurons in different visual areas in the visual hierarchy in terms of the area of these rectangles here, and then the thickness of these diagrams gives us is an estimate of the amount of connections you have between areas. And I think it's a useful summary diagram. So a lot is known about the computational functions of feedforward. Certainly a lot of excitement over the last 6-7 years in terms of how deep convolutional neural networks seem to capture some of the aspects of object recognition and its organization in dealing with natural image input. But it's also been known for a long time that we have lateral interactions that are involved in visual computations within areas. And the topic for today is, it's also been known from the connectivity and a long history of research that there is a role for feedback. There are bidirectional connections, and that's illustrated in the next diagram. So I'm going to, again, just to reference those dotted lines there, that I'll be talking about effects that we believe are due in V1 and V2. Just another point to underscore relationship between feed-forward connections and feed-backward connections in both the ascending and descending feed-forward and feedback pathways. One of the things, I'll have a brief opportunity to mention that one of the ways in which people are using fMRI for evidence of feedback is it reflects the anatomical structure of feedback connections, which tend to project backwards to superior, superficial or deep layers of cortex. And so even with human fMRI, you can get some kind of constraint if you have high enough resolution to begin to separate out layers. And I won't talk a lot about that, but I have one little graph to show. Okay, so the key questions of why feedback signals to low-level cortical areas. Today I'm not going to give you any dramatically new ideas. What I hope to do is show you some evidence, experimental evidence from our own labs that is converging on several ideas. There's certainly a long history of results and theory relating attention, visual imagery, and working memory more recently effects early on in the visual system, early on in V1, for example, and V2. Probably from a computational perspective in terms of fleshing out potential reasons that are testable and of interest are learning this idea of building or tuning network hierarchies for frequently needed tasks. And then also sorting through which unresolved low-level features may belong to higher-level hypotheses, if you will. A problem that's whenever you have complex, ambiguous images locally, how do you resolve these ambiguities if you don't have enough information in the bottom-up flow? And in that context, I'd like to focus on three sets of experiments that address this question of whether we can understand feedback signals in terms of ideas that have been around for a while. One is predictive error coding. Another one is binding across levels of abstraction. And finally, this idea that's probably less well known, and that is that the idea of consulting low-level cortical expertise. And all of these are somewhat vague ideas, and hopefully as the field progresses, we'll have a better idea of how to test these ideas and distinguish them. And I'd like to also focus on processes that are probably automatic, at least in part, rather than feedback effects or top-down influences that may have more conscious direct control. So just a little bit of history. So my own interest in nature of feedback came from early work on Bayesian models. Some of that was inspired by work on regularization theory here at MIT in Poggio's lab. And there's this idea that if you want to make given an image or giving some data, if you want to make good bets about the underlying causes or explanations, S, scene properties S, you could use Bayes' formula to break up the constraints into a likelihood and a prior term. And although these terms don't necessarily have to be embedded in mechanisms, you can imagine them, or at least dynamic processes, they could be part of a feed-forward structure. This whole idea of being able to test a prediction, given enough hypothesis S against the image data coming in as a measure of error, was intriguing to me. And so a lot of the early inspiration came from this idea in the 1980s that you could think about vision as solving an inverse optics problem, where you had some external world property S produces an image, and you have the brain's representation. And if you had some kind of generative model, you could also run the inverse of your forward model back to test and see whether your image representations are capturing a correct interpretation. Partly for fun, I thought, maybe that's at my time of life where I'm thinking about, we worry about people forgetting the contributions of earlier generations. And a generation before me was Donald Mackay. Some of you may be familiar with his son, David Mackay, who passed away a few years ago. And this was a paper from 1956. And which he wrote, it's been suggested that the correlated perception as distinct from reception is activity which organizes an outwardly directed internal matching response to signals from receptors. This organizing activity amounts logically to an internal representation of the feature in the incoming signals to which is adaptive. i.e. the feature which is thus perceived. And I thought that was interesting, sort of captures this idea of the use of a generative model for testing early level hypotheses at the input level. And even went on to talk about hierarchical structure. It's postulated wherein much of the organizing activity is concerned with modifying the probabilities of activity. So very general statement, but it reflects, I think, a program of research that many of us are doing and are interested in. So today I'm going to talk both about psychophysical and neuroimaging approaches to addressing the question of feedback, which is at a very coarse scale. So it's going to necessarily involve some basic assumptions, which may or may not be true, but you have to start somewhere. And first of all, the idea is the experiments we're going to talk about will take advantage of the hierarchical feed-forward structure of visual cortex. And so that's the idea that if you've got at the input, we've got receptive neurons with narrow receptive fields that effectively have tunnel vision. They feed forward and gradually accumulating information. And eventually that we have information that's sort of summarized at a high level that captures a wide range of scale by these gradual sequential processing stages. But the idea is if we have measurements or ways of inferring activity early on that's distant from where the local direct stimulation is coming from, then we have a way of measuring effective context. And so the idea is to present a stimulus over here, and then we look at contextual modulations. The second way, which I alluded to a little while ago, is that one can also look for context-dependent modulations of activity in superficial or deep layers of cortex. Some caveats that are worth keeping in mind. I mentioned already before that we know we've got lateral connections, and so one has to argue or at least consider the possibility that these contextual interactions that you might measure at an early level are due to propagations within area or within a level of abstraction. And then also, I often think of as the elephant in the room. I gave a talk once some years ago, and Ray Guillory, the second author, accused me of being corticocentric because I was only talking about effects in V1 that might come from the cortex itself rather than from the thalamus. But there's a sort of rich body of evidence for thalamic cortical interactions. So these are caveats, and the hope is that our interpretations will last despite these caveats. So I'd like to start a long time ago with work that was first done with Scott Murray and then later on with Fong Fong. It got me interested experimentally in the question of predictive coding kinds of explanations. And this was an experiment that was done in which we took a look at this called translating diamond stimulus. And if you look at the one on the far left there, we won't take time for you to experience it, but one of the interesting properties that it seems to be bistable. And you can either see a whole shape or it breaks up into local individual shapes. And when we looked at fMRI activity, in early visual system, we found that the response to when the person perceived a whole diamond, activity in V1 was suppressed. It was as if there was this high-level explanation that was predicting downward, subtracting off the prediction, and you had effectively an error signal that was low. But if the diamond, you didn't have a good perceptual explanation, this was the intuition, then the activity in V1 would go up. Now, one of the problems with this study that bugged us for a number of years was it wasn't clear whether the suppression of early activity was localized in space and feature type. So, for example, we did some measurements where we looked at areas that were really far from these red lines, far from what the areas that were being directly stimulated retinotopically. And we could still see some evidence of suppression going up and down. Our fMRI wasn't that good at localization. And so we were concerned about whether our interpretation was location and feature specific. And the second question was whether this enhancement or suppression of activity that you see early on, how it may depend upon stimulus and task. Like if you have clutter or more information, more background, confusing elements, to what extent is your modulation, contextual dependent modulation, dependent on those contextual features. So we did an experiment. This was an experiment done with Dongjun He and Feng Feng. By the way, Feng Feng there, at that time he was he was a graduate student in my lab, and then he went on to be, he's now executive associate director of the McGovern, his sister institute at Peking University, and the dean of psychological sciences at Peking University. He's on a sled in Minnesota in that picture, so it's a long time ago. Well, we thought we'd do a psychophysical experiment to address this question of whether suppression was localized to early cortical regions corresponding to feature-selective neurons directly stimulated. And so the idea was to take advantage of the known and well-established degree of spatial orientation selectivity in low-level cortical areas, in particular V1 and V2, and the sensitivity to whole forms in higher-level cortical areas. But rather than doing fMRI, we thought, well, maybe we can address this using what Bela Ulez, known for random dot stereograms, called the psychophysicist the electrode, and that's to use psychophysical adaptation. And so we had two stimuli conditions, or two adaptation kind of conditions to draw on. And the first one is the familiar and old observation of the tilt after effect. And so the idea was that if you looking here, we have a vertical grading. It looks vertical to everybody, but then if you were to stare at this tilted grading for a while, a minute or two, and then you go back and look at the vertical grading again, it looks like it's tilted the other direction. And this is known to be fairly specific in terms of orientation and location. In terms of whole shapes, another kind of adaptation effect is an adaptation to the aspect ratio, in this case, of a diamond. So here we have a diamond shape. If you then, it looks at what we call normal, and then if you adapt to a skinny one, so you stare at the skinny one for a while and then go test on the original one, then the original one now looks fatter, okay? So the thought was we can somehow combine these two effects into one stimulus manipulation and try to tease apart whether the adaptation effects on one condition for example, a whole percept versus a local percept yield different adaptation effects. So here's how we combine them. So I've always been fond of this moving diamond stimulus. And so we created a stimulus that looks like this, and it was just gently rotated. The little circles there indicate how pixels in this image sort of just rotated. The idea was to prevent after images to those black and white little grading patterns there, so they get smeared over a small region of retinotopic space. And then introduce two conditions. One condition is which we have these three horizontal uniform field blank bars there. And the idea was to cover up the vertices, and then if you cover up the vertices, the motion that a person sees is of a diamond moving behind three bars. So you integrate all four of those grading patches into one whole, okay? And but if you just extend or just drop these lines a little bit below the edges of these gradings, then these grading patterns look like they're floating in front, and they don't belong to a whole shape anymore. They have four independent bits, or less dependent bits. And so we've got these a diamond percept, and then we've got four oriented patches percept that are not grouped together into one coherent object. And so just jumping ahead in the results, so we could measure both a tilt after effect here, and we could also measure a shape after effect under these two conditions, whether a person perceived the stimulus in terms of a diamond or non-diamond. And we got this nice interaction effect here, in which the under the non-diamond percept, we get a big tilt after effect and a reduced one when we're seeing the diamond. And conversely, if you look at the shape after effect, we have a smaller shape receptor effect for the non-diamond percept and the big one for the diamond. And so our interpretation was that this perceptual grouping, when you experience the diamond, reduces the adaptation to local tilt, which we presume is affecting early level neurons, and presumably preferentially V1 and V2. And there are a number of other manipulations we did. You can even get adaptation across the eyes, suggesting that we've got both an adaptation to monocular neurons and binocular neurons in this stimulus. So we took heart. This was consistent with the idea of local, lower level feature-specific suppression, akin to that idea of predictive error coding. The next study that I'd like to tell you about addresses this question of just a little bit of background. There's a fairly large literature that has looked for these context-dependent effects on either an increase in activity measured through fMRI or a decrease measured through fMRI as a function of how elements get grouped. And so there are some conditions under if you're trying to group elements, such as in the stimulus I have here, where we've got these little Gabor elements that are in sort of circular alignment, you get an increase in activity. There are other experiments that also show an increase of activity when you have groups of elements that are in clutter. And but there are also studies, for example, that show conditions under which you get suppression, like the experiments that we have. And it's been a puzzle as to can you predict when you might expect an increase in activity or decrease in activity in early cortical areas as a function of different stimulus conditions. And so one of the ideas that we thought we'd address, and this was done with a graduate student, Chung Chu, who's now working with Andrew Stocka at University of Pennsylvania, and Professor Cheryl Ullman, who's in my department at Minnesota, where we had this idea that the enhancement that's sometimes seen at early levels when you're perceptually organizing something may have to do with how much clutter, how much separation is required by the visual system to pull out the form in the presence of distractor elements. And so in this experiment, we had the four conditions shown here. We have what we call the aligned condition, where all these little Gabor elements are sort of, their orientation is cotangent to some invisible circle. We have unaligned, where we just randomize the orientation. And then we can also have conditions where we embed those aligned circles in the clutter here, and the unaligned in the clutter. And then we have a subject to a task, fixation task, and ask them whether they these Gabor functions are aligned or not. Okay, so here are the results. Oh, I should mention too that we ran localizers so we could analyze the data coming from voxels that were directly stimulated within that red annulus, as well as ones on the outside. And so we could pull apart the target regions from the background regions. And the main effects we got were in the target regions in V1 and V2. And so the main story here, so this is the bold response difference. If we've aligned elements compared with activity in unaligned elements in the background, that's when we got the boost in activity. So unlike the suppression results that I showed when we had that diamond, we've seen an enhancement of activity in the presence of these background elements here. I see that both for V and V and the V2 voxels. We had some evidence for hinting at suppression of activity in this no background case. That corresponds to the top two conditions there, but it's not a strong effect. The big effect for us was this effect of background that seemed to amplify the activity associated with grouping these elements, these Gabor elements in the circle. So summary: in background clutter, V1, V2 activity in that target region increased for aligned versus unaligned features. Chung Chu went on to also do an analysis of the correlations between the time series in V1 and seed regions in V2. And this is shows some of the results. So we did several different analyses. This is a psychophysiological measure which compares the time series in the space of the neural activity, where you deconvolve your fMRI time series and you can compare that, cross-correlate that with the conditions, the 4 conditions we have here. And the basic result there is summarized in this diagram, where the strongest correlations we had corresponded to this condition, where we compared the aligned with unaligned in background. So in a nutshell, the idea is we have an increased correlation between V2 and V1 voxels under those conditions when a person is organizing the aligned elements in the presence of this background as compared to the unaligned ones. Okay? So we interpret that as increased coordination, if you will. The analysis metrics, so if you use a seed region in V1 and analyze V2, you get the same kind of story. So it doesn't tell us about the causal relationship in terms of timing. None of this really does, but it does suggest that this idea that there's an increased role for feedback between areas when you're trying to organize ambiguous elements, potentially ambiguous elements in clutter, that activity goes up. So there's the summary. So measures of functional connectivity between V1 and V2 increased when perceiving aligned versus unaligned contours in background. Here's another study that was done with Damien Mannion and also Cheryl Ullman. It's also consistent with this general idea that if you have a complex, potentially complex scene that requires integration of locally ambiguous bits, that you might see increased activity when those things belong together, sort of in the opposite direction of predictive error coding. So in this stimulus, the idea was to, I'm going to keep pointing it over here, was to select a number of apertures that would hide most of the scene. So it's like you're looking at this natural image scene through a set of holes. And if you adjust things right, you get, and with a little bit of practice and subject, you can see through the holes. So you've got the holes there, and you can organize that whole scene behind the holes. And the experience is just like you're looking through holes. So that's where you're well organizing these patches that you see through the holes. But then you can take another scene and pick out patches that don't belong with the first one, and insert them or not insert them. And you can leave the original patches, or you can insert the incoherent or incompatible patches in these apertures in the scene. And so this would be, this is an example here where there's some patches that just don't fit with the rest of the patches. They don't belong to that scene because they came from somewhere else. And so now the question was, if one localizes voxels, retinotopic voxels at these locations, do we see an increase in activity or decrease in activity for those patches that belong or don't belong to that whole scene? And so the answer here is rather simple, and that is that we found that there was, if the patches were coherent, in other words, actually belonged to the scene, then though the localized activity there did go up relative to the other patches. So this is also consistent with that idea of boosting activity if it belongs together. You've got the whole scene interpretation, and you've got activity early on in cortex that seems to be boosted in some sense, given the consistency. And this was a paper published in 2015, and this is the only layer analysis that I'm going to talk about, and the only one that we've done. But I thought I'd just point it out because at least it's suggestive, despite I think a lot of caveats that have to do with this kind of analysis. So this was done at higher resolution than the other studies, and this, what we have here is on the x-axis is the distance from the white matter. And we have a measure of the coherent minus the incoherent response as a function of layer. And we do seem to see increased activity closer to the superficial layers of cortex. So consistent with this idea that we might be picking up a signal that's being fed back to superficial layers. Anyway, I'm always a little hesitant about interpreting this along this, just because I know of all the problems that we've had in actually being confident about layer specificity and its relationship to neural activity versus vascular. Okay, so the summary so far is that from the adaptation experiments, we have evidence of local orientation-sensitive activity in early visual cortex, and as a consequence of high-level perceptual organization, as a consequence of seeing that whole diamond shape. And so just from adaptation, we have results consistent with this predictive error ideas. But we also have evidence for enhancement under conditions of detecting feature alignment in clutter, consistent with this idea of binding across levels of abstraction, binding across levels of cortical hierarchy, and certainly in V1 and V2. And this figure sort of summarizes the general ideas. If you don't have a lot of clutter, then a high-level model such as a circle can do a good job at fitting elements that by themselves aren't naturally grouped. But if you take that same stimulus there and you embed a bunch of other elements, then you lose that coherence. But you can get it back again if you happen to have stimulus conditions where you can locally group based on similarity and proximity, and then you can solve this problem of figure background segregation. Okay, so we've basically I've showed you evidence on relating to the idea of predictive error coding, which is one way of sorting through potential features that do belong or don't belong with a high-level perceptual interpretation. We looked at this idea that I looked for evidence of binding across areas where a high-level model links together low-level information at a different level of abstraction. And so the third idea for what feedback may be doing, what its function may be, I'm going to call consulting lower-level expertise. I know it's rather vague, but part of the problem is we don't have, I think, a really good ideas yet of how to interpret a small set of results that come from a number of labs, and in particular, the result I'm going to talk about here. But the general idea is that foveal V1 is an expert in processing fine spatial detail and orientation information. There was a lot of real estate there I showed you in the first in that wiring diagram from Wallish and Mavshin, both in V1 and V2. And in particular, we also know that so the fovea has a lot of real estate. The foveal area, V1 itself, has a lot of real estate, of course, devoted to spatial processing. And so to look at this idea, this general idea that maybe feedback, maybe areas of cortical areas have expertise in ways that we don't normally think of, seems to be, we can address this using this general idea and explore it in the context of V1. And I'll talk a little bit more about the interpretation later, but this idea of expertise at a lower level, it's not a new idea. Hisser and Huckstein and Lee and Mumford had similar ideas with the idea that V1 might be an expert at a spatial buffer. It's an expert at dealing with fine detail. So the question is, are foveal cortical neurons consulted for the fine-grained spatial analysis of details? And here's another domain in which I was inspired by work here at MIT when Mark Williams was in Nancy Kanwisher's lab. So a little bit of background here. So in 2008, Mark Williams, based on research here, published this paper in which they did a multivariate pattern analysis of voxels that corresponded to a foveal region that wasn't directly stimulated, while subjects made a decision about whether this object up here, spiky smoothies and cubies, whether this spiky object here was the same at a detail level as in the lower left. So the same category, but they differ slightly because their little spikes might not be arranged quite well. And the interesting finding was that the voxels in these non-stimulated areas could be used to decode the category of the object that the person had been exposed to. In other words, to say, just looking here, can you read out from foveal voxels, foveal stimulated voxels, whether it was spiky, smoothie, or cubey? And when I was talking to Nancy earlier today and talking about when this first came out, we thought there has to be some kind of behavioral correlate of this. Does it actually affect behavior? And together with Damien Mannion, I mentioned earlier, we tried a number of things. They didn't work. And so I gave up temporarily, but I'm going to revisit a method that does work shortly. And partly that was inspired by subsequent work done by Mark Williams, in which showed that there was a behavioral effect. And they could get a behavioral effect by using a transcranial magnetic pulse stimulated to foveal cortical regions in the back of their subjects' brains at the right time. If you did it at the right time, you could disrupt their ability to do this task. And that time it was on the order of 350 to 40 milliseconds after stimulus onset. So keep that number in mind. I'd rather not use TMS if I don't have to. I'd rather not use fMRI if I don't have to. And so we tried some physical experiments. And along with two other groups, we discovered that one didn't need to use a TMS pulse to get the good interference or have a disruptive effect on your ability to make fine-scale discriminations of these patterns. And the idea is to present the stimuli and then flash a noise pattern up, right, where you'd expect to activate those foveally sensitive voxels from early all the way into V1 and V2. And the question was, if you insert that mask, really don't call it it's not really overlapping the stimulus. If you interfere, it stimulates this noise pattern at the right time, can you disrupt performance? And so I mentioned there were a couple of groups that, including one that worked later with Mark Williams, Weldon et al., who showed you could get these interference effects in fovea with just a noise stimulus. Well, I had sort of complained to my colleague Sheng He that I had struggled along with Damien to get some kind of behavioral effect ourselves. This was before I started using this noise technique. And so my main contribution to the rest of this talk here actually was to tell Sheng I got null effects and I couldn't get a big effect. And Sheng went and worked with a student at the Chinese Academy of Sciences, and they came up with a paradigm that has produced a number of interesting results, which I'd like to tell you about now. So the previous studies hadn't addressed three, I think, key interesting questions that we thought we would use this noise technique to address. And the first one is foveal processing only engaged for tasks requiring fine spatial detail, because that's the basic task that had been done so far. A second question is, if there's this disruptive effect, can we get an estimate of its temporal window? How long does it last? And then thirdly, is the feedback deployed automatically when the task requires it? And if I have time, there are a few other interesting questions I may bring up too, but let's focus on these now. So here's the basic paradigm. And so we've got, in terms of timing, the fixation mark comes on, stimulus comes on with these two in this case, two spiky objects. Remember, the task of the observer is to say whether they're same or different images, not same or different categories, same or different images at the image level. And then that's followed by one of 5 different delays in terms of when the mask gets flashed on. The mask is very brief. It's only 83 milliseconds. And then the observer makes a response. So first of all, the basic observation of a disruption we replicated, and here's just take you through this diagram here. We've got measure of D prime, how well people are making this decision of same or different, as a function of these different noise onset times there. And the dashed line up here is the control condition to see what they did and measure their performance when you didn't have this interference flash. So at 50 milliseconds, we get a big suppression, and in a way that's not so surprising. It's consistent with attention to action. The stimulus is actually still on when that flash comes up, and so we'd expect some kind of interference. You generally get fairly global effects of a flash based on distracted attention. But then within 150 milliseconds, performance goes up. Interestingly, we get this drop, 250 milliseconds. Okay. So a couple things we can infer from this. One is that it looks like we did some modeling, assuming this was a tensional effect, and this depression here was an effect of the feedback to foveal cortex, and we get a fairly, I'll show you some curves later, but you get a fairly narrow time window of effectiveness. It's on the order of about 130 milliseconds where we can get this interference effect. The second thing you can remember, I mentioned earlier the MS results from Mark Williams' lab about you get interference on the order of 350 to milliseconds or so. Why is ours 250? Well, it's actually not inconsistent, because with the TMS pulse, you have to give the system longer time, because with TMS pulse you can, you're applying it and have to feed them, take the time for the mask, for the center noise mask to get through. So sort of in the ballpark, 100 millisecond difference or more. Might be a little bit long, but at least it's not drastically inconsistent. So a narrow time window of about 130 milliseconds. There's fine detail required to get the effect. So in order to do that, we low-pass filtered these objects. I should say Zhu Fan did it. I didn't actually low-pass filter them. And now we get a different pattern of results where performance is initially suppressed again, presumably due to this global attention distraction process, and then performance gradually goes up, but we don't see any dip in this curve, suggesting that the effect we're getting has to do with the task of now analyzing fine spatial detail. Again, consistent with this general intuitive idea that we're somehow tapping into the expertise in the fovea of dealing with its ability to deal with fine detail, even though there's no stimulus there. And the question is, what's going on? So no corresponding drop for low filtered images, and I won't show you the data, but we also did a number of other conditions were tried, including speed discrimination task, equally difficult. Dots moving in one direction in the upper right, another direction lower left, and you don't get you get a pattern similar to this blur condition here.
Do you see, have you tried a condition where you have like an annulus of noise rather than a just center
Yes.
of noise?
All right, great.
Do you not see the effect?
Great question. Yeah, you don't see the effects if you put the noise on the outside. Yep. We've got a graph on that, but that's, yes. So the noise has to be in the middle. Yeah.
Does the high spatial frequency to be task relevant for you people?
didn't directly test that. The presumption is it does. But yeah, I mean, that would be another condition to directly check. I mean, there's an interesting point to make here, and that is that in order to presumably recruit or at least anticipate the need for foveal processing, how does the visual system know it needs it if the stimulus is already sort of blurry in the periphery? One possible answer for that is that there's sufficient high spatial frequency information for the system to know it's high spatial frequency. It just, you have probably perhaps related to crowding. And so it recognizes high spatial frequency. There's a job for the fovea to do. And so let's prepare the fovea for that kind of analysis, where you don't have the problems of clutter, at least not to such a large extent. Okay, is feedback deployed automatically or only when the task requires it? And in full disclosure, when Sheng told me he was going to do this experiment, I thought, wow, it's cool, I'm jealous, I wish I had thought of it. I hope you find it the same as well. The idea was to take advantage of a well-known result, going way back to Sheplard and Metzler, and these are mental rotation results. And that was the idea. If you have to decide whether two objects are the same or different, the amount of time to do it with the criterion level of accuracy depends upon the angular separation between the two, suggestive of some kind of analog process in the brain where you're mentally rotating one object until it's aligned with the other, and then you make your decision. Okay, and if they start off with a big angle, it takes you longer to do the rotation. If a small angular difference between the two, you can do it faster. So the idea was to incorporate that into this experiment with the interference patches, and so that to have conditions in which we, as we saw before, where this patch there has to be compared with that patch down there, and they both have the same orientation. And then also have a condition where the alignment is different, okay, but only by, I'll show you how many degrees in a minute, I forgot. And then one that requires even more mental rotation, if you will, bigger difference between the two angles that separate those two. And so in this task, it was the same task that subjects had before, except they were told to fixate and take the time to mentally rotate until they had them in sort of mental correspondence with each other, and then make the decision. But as before, we try to disrupt the performance by inserting this noise patch in the fovea, away from where the stimuli actually are, and look at whether have a decrement in performance, and if so, at what times. So here are the results, the same kind of plot we saw before. We have D prime on the vertical axis, and we've got our different stimulus onset asynchronies plotted on the bottom. And the top one, we basically replicated the first experiment. This is where I showed you the curves where we can fit an attentional factor and an interference factor to get that narrow time window I mentioned earlier. But we get this depression or suppression interference effect at 250 milliseconds. However, if the subjects are looking at the stimulus where we've got a 40-degree difference, so they now have to take a little bit more time to do the mental rotation, then we see we've delayed the period at which we get this maximum interference. And if they have to do an even bigger rotation, mental rotation, then we can get an effect of the delay. It's almost a half second. So it's a rather long period of time in order for this effect to occur. Okay, so just to summarize, the basic results were that when mental rotation was required as part of this peripheral object discrimination task, the temporal window shifts the time that foveal noise disrupts the peripheral object discrimination. And so this is mental rotation is consistent with this idea that retinotopic cortex is not automatically engaged at a fixed time following the stimulus presentation, but is recruited, in effect, or invoked when the rest of the brain, if you will, is ready to do that processing. So I'm not going to take time to talk about in detail there are several experiments in this study, but I did want to take time to summarize the results because I think they may be key to understanding what's eventually going on. And in a further experiment, we examine the question of whether this foveal interference depends upon saccade planning. Now, it's a little more complicated experiment, but the answer is yes, it seems to. In other words, if you have a task where the subject the part of their task is to fixate away from the target. You have a spiky object up here, and they have to make a rapid fixation down there. But if you insert this interference pattern before they make the saccade, we lose this suppression effect. So it's as if saccade planning seems to be part of it. It's a little bit mysterious, so I don't think that can be the whole story. But it does still consistent with this idea of the visual system knowing about its own expertise, if you will, at a lower level and preparing a system for an analysis of fine detail. And we also did fMRI experiments in which we looked at whether information not only about the category could be recovered from the non-stimulated voxels, as has been shown by Mark Williams in Nancy's lab, but whether the image property of the overall orientation, whether this QB or this spiky was oriented horizontally or vertically. And you can recover both kinds of information from those foveal voxels that aren't directly stimulated. I think I've summarized everything so far, and I wanted to leave you with sort of just one set of questions I have that go back in my own history. In fact, I might have even talked about these results at MIT maybe 12 years ago or so, and something I've been puzzling about for a long time. And revisiting, this is an older finding in which involved the perception of the perceiving of the size of an object. So hopefully for most of you, this ring at the back of this hallway looks a little bit bigger than the front. So you can make these effects really big if you have the right cues. And a good example of that demo comes from Goldstein. I don't know if any of you have seen this before, but this marmot railway track, they both have the same image size, okay? And if I move that image, I'm not doing anything in terms of scaling the image, but we can fit that bottom image right on top of the other. This is a really, really strong effect. And with work done with Scott Murray and then subsequent studies with Funk, we looked at whether this effect is manifest at all in primary visual cortex in terms of the spatial extent of activity. So we know from retinotopy that if a ring actually does get bigger, then you're going to have a spread of fMRI or bold-related activity go from representations that are more posterior to ones that are more anterior. And we found that you also get that increase in spatial extent of activity if it's just an illusory size change. And one reason I wanted to bring this up, because I know there are a lot of bright young people here, and I've never quite figured out what this means. And it really, really puzzled us. And so one question, so this just summarizes the results, and I just want to wrap up here quickly, but I wanted to leave you with a few questions that might be related to this idea of the cortex, visual cortex, as using feedback because of its expertise, if you will, its expertise in spatial processing of things like size. And so what does that increase in perceived size result? Why does it result in this increase in spatial extent of activity? And one hypothesis is that 3D object size is represented by the extent of activation, V1. And I think there are lots of reasons why I don't think that's the case. V1 could be used at some more abstract levels, a kind of sketchpad to make relative comparisons. But then there's this third idea, and that is that these illusions persist even if you have a hard segmentation problem. So I don't know if this works for you, but this ball here, for me, still looks bigger than the one below, and you can do quite a few manipulations of that sort. So there's a challenge just in terms of deciding which bits and pieces belong before you, just in terms of angular extent, before you decide how big is the object and you incorporate 3D depth. And so that, this is again speculation, but maybe that's somehow related to this idea of V1. We're getting these feedback effects in V1, these contextual effects in size, because of a process that needs to pull out really what is the angular extent. And after all, if we do have a written topic representation in V1, that's a reasonable place to get that angular estimate. But then we still need an explanation for why we have a bias. Why is there this error in angular extent? Okay, so I need to wrap things up here. So I presented some evidence that one of the functions has to do with the selection and integration of local features through feedback. We looked at reduction of local activity related to explained higher level features, higher level properties in terms of predictive coding, but also that show that increases in early activity depend upon the complexity of the segmentation problem, the background clutter and so forth, suggesting a role for binding across levels of abstraction. And finally, this idea that evidence to support this idea that V1 may be involved in a richer set of computations than just spatial filtering or spatial temporal filtering, but it's also involved in high resolution tasks that are recruited when the task demands it. And this open question here of whether our results are also related to predictive remapping related to saccadic planning. And maybe I'll just skip to the end here and say just a reminder that everything I talked about here owed a lot to other people. So thank you.